Hello everyone, I'm Uddhab Karki. I'm doing PhD here in Arkansas State University in the Molecular Bioscience Department under the kind supervision of Dr. Xu. Today I'm going to present part of my PhD dissertation work. In this project, I'm trying to produce cell wall deficient plant cells to enhance the biotherapeutic protein production. I use the tobacco plant and make the callus from this plant, and I try to remove the cell wall, which will enhance the biotherapeutic protein production in the industrial scale. Now moving towards the research background. Those therapeutic materials produced by using the biological means, including change of the DNA, is called biotherapeutics. Human insulin is the first FDA-approved biotherapeutics. It has a great global market and is very costly. More than $1 billion will be needed to make a new drug from concept to the market. Animal and bacterial cell culture are two major production platforms. But still there is some challenges in animal and bacterial cell culture system. Animal cell culture system is costly, time consuming, and there is a possibility of cross contamination. Meanwhile, bacterial system can produce the complex humanized proteins. So plant can be a suitable alternative production platform because it's a cost effective, easy to scale up, safe to use, and can be produce complex proteins. Protalix Therapeutics is a commercially successful example of plant therapeutics. They produce glucocerebrosidase in the carrot and tobacco cells, which is the first FDA-approved plant-produced therapeutics used for the lysosomal disorder. But still there are some challenges in plant production platforms, like low yields, formation of the big clumps, having the large vacuoles which have the more water, and it makes it difficult for the long-term storage, unlike in animal cells. They have some proteolytic enzyme that can degrade our target proteins. So I think cell wall removal is a promising solution. Now talking about the plant cell anatomy. Plant cell has the cell wall and the large vacuoles, and the cell wall components is made up of the pectin, hemicellulose, and cellulose. They form the interconnecting structure and give the rigidity to the plant cell in the environment, natural environment. Now let's discuss about how modern technology can remove the cell wall permanently. In this system, there is a pre-designed single RNA which leads to the target of our sequence and DNA cutting enzyme called Cas9. They will form the RNA protein complex and transport it to the target DNA sequence in the chromosome. They will make the target gene permanently non-functional. This technology is called CRISPR-Cas9 technology. Now moving towards the research plan. I use two approaches in the research. First one is chemical inhibition approach, and the second one is gene modification approach. At first, I treated the plant cells with a cellulose biosynthesis inhibitor dichlorobenzonitrile and observed the protein secretion, its morphology, and I did the characterization. In the second approach, I used the modern technology which I already mentioned to the target gene knockout. In this system, I use the bacterial system for the transformation of this Cas9 and the guide RNA of my target to the plant cells, and I did the characterization of the transformed cells. Let's talk about result. From the chemical inhibition approach, I found that cells are alive, divide slowly but formation of the large clumps, which was disappeared after treating the pectin hydrolyzing enzyme pectinase. So these clumps may be due to the presence of large amount of pectin, and there is normal production and secretion of reporter proteins. What I conclude from this result is that there is no vital role of the cellulose in the plant cell viability in the controlled environmental condition. In the gene modification approach, I successfully knocked out the 115 base pair of cellulose synthase A gene, which is responsible for biosynthesis of cellulose in plant cell wall. But still they are in the mixed form with the wild type. So I'm screening the pure knocked out lines. After selecting the pure knocked out lines, I will characterize its anatomy of the cell wall and I will check the function of the cells. So I'm targeting the more site of these same genes. That's why I can make it more non-functional. That's why it can lose its functionality permanently. So I can get the cellulose-free plant cell. I'm extremely thankful to my advisor, Dr. Xu, my committee members, lab members, Arkansas Bioscience Institute and MBS program, Arkansas State University, and all the other funding agencies. Thank you for listening me. Stay safe.